Welcome to the new video MCQs on microbiology. Question number 1. Which bacterial component determines antigenic specificity? Ribosome, capsule, cell membrane, cytoplasm. The right answer is option B, capsule. Capsular polysaccharides are major antigenic determinants. Question number 2. Which structure is responsible for bacterial motility? Pili, ribosome, capsule, flagella. The right answer is option D, flagella. Flagella are long whip-like structures that help bacteria move. Question number 3. The main function of the bacterial capsule is to produce toxins, provide nutrition, protect against phagocytosis, aid in respiration. The right answer is option C, protect against phagocytosis. Capsules are antiphagocytic and enhance bacterial virulence. Question number 4. Which staining technique differentiates bacteria based on cell wall structure? Gram stain, Albert stain, Ziehl-Neelsen stain, Giemsa stain. The right answer is option A, gram stain. Gram stain differentiates bacteria into gram-positive and gram-negative based on peptidoglycan thickness. Question number 5. Which bacteria lack a cell wall? Mycobacterium, Chlamydia, Mycoplasma, Rickettsia. The right answer is option C, Mycoplasma. Mycoplasma species lack a cell wall and are resistant to beta-lactams. Question number 6. The normal shape of cocci arranged in chains is called staphylococci, diplococci, tetrads, streptococci. The right answer is option D, streptococci. Streptococci divide in one plane forming chains. Question number 7. Which medium is both enriched and selective? McConkey agar, blood agar, nutrient agar, chocolate agar. The right answer is option A, McConkey agar. McConkey agar is selective for gram-negative bacteria and differentiates lactose fermenters. Question number 8. Lactose fermenting colonies on McConkey agar appear colorless, black, green, pink. The right answer is option D, pink. Lactose fermentation produces acid, turning colonies pink. Question number 9. Which enzyme neutralizes hydrogen peroxide in bacteria? Coagulase, catalase, oxidase, urease. The right answer is option B, catalase. Catalase breaks hydrogen peroxide into water and oxygen. Question number 10. Which bacteria are acid-fast due to mycolic acid? Streptococcus, Escherichia coli, Mycobacterium, Vibrio. The right answer is option C, Mycobacterium. Mycolic acid in cell wall makes Mycobacterium acid-fast. Question number 11. Which gas is required for the growth of obligate anaerobes? Oxygen, carbon dioxide, nitrogen, none. The right answer is option D, none. Obligate anaerobes cannot tolerate oxygen. Question number 12. The optimal temperature for growth of most human pathogens is 25 degrees Celsius, 30 degrees Celsius, 37 degrees Celsius, 45 degrees Celsius. The right answer is option C, 37 degrees Celsius. Most pathogens are mesophiles adapted to body temperature. Question number 13. Endospores are best detected by spore stain, acid-fast stain, capsule stain, gram stain. The right answer is option A, spore stain. Spore staining, for example, Schaefer-Fulton, specifically demonstrates bacterial spores. Question number 14. Which form of gene transfer requires direct cell-to-cell contact? Transformation, conjugation, transduction, mutation. The right answer is option B, conjugation. Conjugation involves transfer of plasmid DNA via sex pili. Question number 15. Plasmids are best described as chromosomal DNA, RNA molecules, extrachromosomal DNA, cell wall components. The right answer is option C, extrachromosomal DNA. Plasmids are circular extrachromosomal DNA with accessory genes. Question number 16. Which sterilization method is suitable for heat-sensitive liquids? Filtration, dry heat, autoclaving, flaming. The right answer is option A, filtration. Filtration removes microbes without using heat. Question number 17. The principle of autoclaving is based on dry heat, moist heat under pressure, radiation, chemical action. The right answer is option B, moist heat under pressure. Steam under pressure increases temperature and kills spores. Question number 18. Which indicator confirms effective autoclaving? Phenol red, litmus, Bacillus subtilis spores, Bacillus stereothermophilus spores. The right answer is option D, Bacillus stereothermophilus spores. These spores are highly heat resistant and used as biological indicators. Question number 19. Which disinfectant is most effective against spores? Alcohol, phenol, glutaraldehyde, chlorhexidine. The right answer is option C, glutaraldehyde. Glutaraldehyde is a high-level disinfectant effective against spores. Question number 20. Bacterial growth curve phase with maximum metabolic activity is lag phase, log phase, stationary phase, decline phase. The right answer is option B, log phase. During log phase, bacteria divide rapidly and are most active. Question number 21. Which bacteria grow best in increased carbon dioxide concentration? Capnophiles, anaerobes, aerobes, psychrophiles. The right answer is option A, capnophiles. Capnophiles require increased carbon dioxide for growth. Question number 22. Which staining method demonstrates metachromatic granules? Gram stain, India ink, Ziehl-Neelsen stain, Albert stain. The right answer is option D, Albert stain. Albert stain highlights metachromatic granules. Question number 23. Which structure helps bacteria attach to host cells? Flagella, pili, ribosomes, spores. The right answer is option B, pili. Pili aid in adhesion to host tissues. Question number 24. Which term describes bacteria that thrive in cold temperatures? Thermophiles, mesophiles, psychrophiles, halophiles. The right answer is option C, psychrophiles. Psychrophiles grow optimally at low temperatures. Question number 25. Which component is absent in prokaryotic cells? Mitochondria, ribosomes, cell membrane, cytoplasm. The right answer is option A, mitochondria. Prokaryotes lack membrane-bound organelles like mitochondria. Question number 26. Which bacterial cell wall component is responsible for endotoxin activity? Teichoic acid, lipoteichoic acid, peptidoglycan, lipopolysaccharide. The right answer is option D, lipopolysaccharide. Lipopolysaccharide, especially lipid A, acts as endotoxin in gram-negative bacteria. Question number 27. Which of the following bacteria shows pleomorphism? Streptococcus pyogenes, Corynebacterium diphtheriae, Escherichia coli, Neisseria meningitidis. The right answer is option B, Corynebacterium diphtheriae. Corynebacterium diphtheriae shows variable shapes and club-shaped forms. Question number 28. Which medium is used for transport of stool samples suspected for enteric pathogens? Carey-Blair medium, alkaline peptone water, selenite F broth, nutrient broth. The right answer is option A, Carey-Blair medium. Carey-Blair medium preserves stool pathogens during transport. Question number 29. The function of teichoic acid in gram-positive bacteria is protein synthesis, DNA replication, cell wall rigidity and antigenicity, capsule formation. The right answer is option C, cell wall rigidity and antigenicity. Teichoic acids provide structural support and antigenic properties. Question number 30. Which bacterial enzyme is detected by the oxidase test? Cytochrome oxidase, catalase, peroxidase, dehydrogenase. The right answer is option A, cytochrome oxidase. Oxidase test detects cytochrome C oxidase activity. Question number 31. Which bacteria are non-motile? Vibrio cholerae, Proteus mirabilis, Klebsiella pneumoniae, Pseudomonas aeruginosa. The right answer is option C, Klebsiella pneumoniae. Klebsiella species are characteristically non-motile. Question number 32. Which method is used to demonstrate bacterial motility? Gram stain, hanging drop preparation, acid-fast stain, capsule stain. The right answer is option B, hanging drop preparation. Hanging drop method allows observation of true motility. Question number 33. Which type of culture medium indicates sugar fermentation with gas production? Blood agar, Robertson cooked meat medium, Lowenstein-Jensen medium, sugar fermentation broth. The right answer is option D, sugar fermentation broth. Sugar broths contain indicators and Durham tubes to detect acid and gas. Question number 34. Which bacteria require intracellular growth for survival? Staphylococcus, Chlamydia, Klebsiella, Clostridium. The right answer is option B, Chlamydia. Chlamydia are obligate intracellular bacteria. Question number 35. The function of bacterial ribosomes is DNA synthesis, cell wall synthesis, protein synthesis, energy production. The right answer is option C, protein synthesis. 70S ribosomes are responsible for protein synthesis in bacteria. Question number 36. Which sterilization method is used for glassware? Autoclaving, filtration, UV radiation, dry heat hot air oven. The right answer is option D, dry heat hot air oven. Hot air oven sterilizes glassware by dry heat. Question number 37. Which chemical is commonly used for skin disinfection before injections? Alcohol, iodine, phenol, formaldehyde. The right answer is option A, alcohol. Alcohol acts rapidly and is commonly used as skin antisepsis. Question number 38. Which bacteria are called halophiles? Heat-loving bacteria, salt-loving bacteria, cold-loving bacteria, acid-loving bacteria. The right answer is option B, salt-loving bacteria. Halophiles require high salt concentration for growth. Question number 39. Which staining technique uses carbolfuchsin as primary stain? Gram stain, Albert stain, Ziehl-Neelsen stain, Giemsa stain. The right answer is option C, Ziehl-Neelsen stain. Carbolfuchsin stains acid-fast organisms. Question number 40. Which phase of bacterial growth shows accumulation of toxic metabolites? Lag phase, log phase, stationary phase, decline phase. The right answer is option C, stationary phase. In stationary phase, nutrients deplete and toxins accumulate. Question number 41. Which culture medium is used for anaerobic bacteria? Nutrient agar, blood agar, McConkey agar, Robertson cooked meat medium. The right answer is option D, Robertson cooked meat medium. Robertson cooked meat medium supports anaerobic growth. Question number 42. Which bacterial arrangement resembles Chinese letters? Cornybacterium, Staphylococci, Streptococci, Neisseria. The right answer is option A, Cornybacterium. Cornybacterium shows angular Chinese letter arrangement. Question number 43. Which bacteria can survive extreme environmental conditions due to spore formation? Escherichia coli, Bacillus, Neisseria, Shigella. The right answer is option B, Bacillus. Bacillus forms endospores that resist harsh conditions. Question number 44. Which component gives rigidity to bacterial cell wall? Lipid A, lipoprotein, polysaccharide, peptidoglycan. The right answer is option D, peptidoglycan. Peptidoglycan provides structural strength. Question number 45. Which antimicrobial action is shown by phenol? Protein coagulation, cell membrane damage, DNA synthesis inhibition, spore destruction. The right answer is option B, cell membrane damage. Phenol disrupts cell membranes and denatures proteins. Question number 46. Which organism is used as a biological indicator for dry heat sterilization? Bacillus subtilis, Bacillus stearothermophilus, Clostridium tetani, Escherichia coli. The right answer is option A, Bacillus subtilis. Bacillus subtilis spores are used for dry heat monitoring. Question number 47. Which test differentiates Staphylococcus aureus from other staphylococci? Catalase test, oxidase test, coagulase test, indole test. The right answer is option C, coagulase test. Coagulase positivity is a key virulence marker of Staphylococcus aureus. Question number 48. Which bacterial structure helps in genetic recombination? Capsule, flagella, ribosome, pili. The right answer is option D, pili. 6 pili facilitates conjugation and gene transfer. Question number 49. Which agent is most effective against vegetative bacteria? UV rays, alcohol, autoclaving, dry heat. The right answer is option B, alcohol. Alcohol rapidly kills vegetative forms but not spores. Question number 50. Which term describes bacteria that grow best at neutral pH? Acidophiles, alkaliphiles, neutrophiles, psychrophiles. The right answer is option C, neutrophiles. Neutrophiles prefer pH around 7. Thank you for watching the video. Subscribe to the channel to learn more MCQs on microbiology.